Imagine looking down the throat of an animal and seeing just hundreds of massive, sharp, backward-pointing spikes.
Oh, yeah, it's intense.
Right. Like, we aren't talking about small bumps here. These are thick, keratinized daggers that line the entire esophagus. And they're designed to, trap squirming prey and literally wring out salt water like a wet towel.
It's a terrifying visual, honestly.
It really is. You'd think you were looking at the anatomy of an alien from some sci-fi horror film, or I don't know, maybe a biological sarlacc pit, but you're actually just looking at a sea turtle.
Yeah, and it completely shatters that sort of quaint image most people have of these animals.
Exactly.
We tend to think of sea turtles as just, gentle, slow-moving ocean wanderers, standard reptiles that just happen to have flippers. But biologically, they are a masterclass in extreme engineering.
They really are.
They've essentially taken the standard vertebrate blueprint, the exact same one we share with dogs and birds and lizards, and they've entirely inverted it just to survive in a tremendously hostile liquid environment.
Well, welcome to today's deep dive. We are going to completely deconstruct the sea turtle today for you. We want to explore how their bodies solve the impossible physics and, honestly, the chemistry problems of living in the open ocean.
It's a massive topic.
It is. And to do this, we are pulling from a really fascinating stack of sources. We've got highly technical anatomy guides, specifically the National Oceanic and Atmospheric Administration's NOAA's incredibly rigorous manual. It's called The Anatomy of Sea Turtles.
A phenomenal resource.
Yeah, and we are cross-referencing that with digital observations from the Olive Ridley Project. And then, to ground all of this in reality, we are relying really heavily on a gritty, hands-on sea turtle necropsy manual.
Right, the one by Dr. Thierry Emwerk.
Exactly. It's written for biologists operating in some of the most remote, isolated wildlife refuges on Earth.
And that combination of sources is just vital for this deep dive because the NOAA manual gives us the pristine, idealized biological map. It's the blueprint.
Right.
But then the Olive Ridley Project shows us how that machinery actually operates in motion.
Yeah, out in the wild.
Exactly. But the Necropsy manual, that shows us what happens when the machinery breaks down. It forces us to look at the raw mechanics of the animal. The plumbing, the wiring, the filtration systems.
It's the gritty reality of it.
It is, because the intense physical pressures they face from the water column, combined with the chemical toxicity of constantly swallowing seawater, it has forced their anatomy into some radical evolutionary corners.
Radical is definitely the perfect word. And, to even begin understanding how an animal functions with an inflating nose and a spiked throat, we have to kind of strip away all the soft tissue and look at the foundation first.
The skeleton.
Right. The skeletal system of a sea turtle represents one of the most profound evolutionary commitments in the whole animal kingdom. They didn't just adapt to their environment, they fused themselves to their armor.
The structural reorganization is staggering when you really look at it. When we analyze a standard vertebrate skeleton, we break it down into the skull, the axial skeleton, and the appendicular skeleton.
Okay, so let's break that down for people.
Sure. So the skull houses the brain case, the jaws, and the hyoid apparatus.
That's the throat bones, right?
Yeah, that complex of bones supporting the tongue and throat, which in the sea turtle is heavily reinforced to handle the sheer mechanics of swallowing prey underwater.
Makes sense.
Then you have the appendicular skeleton. That includes the appendages, so the flippers, the hind limbs, and the pectoral and pelvic girdles that anchor them all to the body.
Okay, but the axial skeleton is where the sea turtle completely just abandons the standard mammalian or reptilian playbook entirely.
Oh, completely.
Because in human anatomy, our axial skeleton is our spine and our rib cage. And our ribs are flexible, right? They expand and contract with our lungs when we breathe.
Right, they have to.
But in a sea turtle, the axial skeleton includes the carapace, the top shell. The spine and the ribs aren't just housed underneath the shell, they are the shell.
Yes.
The ribs and vertebrae are physically fused into a continuous shield of living bone.
And what's fascinating here is how often people misunderstand that. It's a critical distinction to make, especially when you're comparing them to other armored creatures.
Like a crab, maybe?
Exactly. A crab or a lobster grows a hard chitinous exoskeleton, which is essentially non-living armor that they wear. They wear it, they molt it, they replace it.
Right, they can climb out of it.
But a sea turtle shell is heavily vascularized dermal bone. It's alive. The NOAA manual actually features these incredible images of cleared and stained hatchling loggerheads.
Oh, I saw those. They're amazing.
Yeah, and even at that tiny vulnerable hatchling stage, you can see the initial dermal bone hypertrophy.
Mm-hmm.
The bone along the ribs is already rapidly expanding outwards, preparing to fuse together into a solid dome.
Which is just wild to think about. Imagine if your own ribcage started expanding outward, pushing through your muscles and fusing on the outside of your back.
It sounds like a horror movie.
It really does. You don't retreat into a shell. You are permanently locked inside your own ribcage. You are the shell. And the terminology biologists use in the NOAA manual reflects this bizarre spatial reality too. They map the turtle almost like a submarine.
That's a great way to put it.
Like they use dorsal to refer to the top of the hull, the carapace. Ventral is toward the bottom shell, the plastron. Anterior is toward the front flippers and head, and posterior is toward the rudder, the tail.
And that plastron itself, that ventral bottom shell, is a really fascinating composite structure because it isn't just a flat plate of bone.
Right, it's more complicated than that.
Much more. It incorporates derivatives of both the axial and the appendicular skeleton. The turtle has basically taken elements of its ventral ribs and fused them with bone elements that, in other animals, would actually be part of the shoulder complex.
Oh, wow.
Yeah, so it ties the upper dome and the lower plate together into a single, unyielding box.
Which is an evolutionary trade-off of just epic proportions. I mean, by fusing their spine and ribs into a rigid box, they entirely sacrificed the serpentine flexibility of their reptilian ancestors.
Completely. A sea turtle cannot bend its torso. It can't twist.
Right. But in exchange, it gains absolute, just crushing invulnerability against the jaws of apex predators, like tiger sharks.
Exactly. It's the ultimate defense mechanism.
And the skeletal architecture is so deeply specialized that it even dictates sexual dimorphism, which I found so interesting. The NOAA source points out that in adult males, the sacral and caudal vertebrae, so the bones at the base of the spine going into the tail, they develop these massive dorsal and lateral processes.
Yeah, those bone processes essentially act as heavy-duty anchor points.
Anchor points for what, exactly?
Well, the male requires a much larger, highly prehensile tail to successfully grip the female's carapace during mating. Because they're doing this in the turbulent open ocean. So the skeleton actively remodels itself to provide the leverage needed for those specific, really powerful muscle attachments.
That makes total sense, but locking your entire body into an inflexible bone box creates a massive, immediate physiological crisis.
It creates several, actually.
Right, but I'm thinking specifically about breathing. If your ribs are fused to your shell and your plastron is an unyielding plate of bone, your chest cavity cannot expand. Humans, dogs, even lizards. We all breathe by expanding our rib cages to create a vacuum, dropping a diaphragm and pulling air into the lungs.
Right.
If a sea turtle's torso is entirely rigid, how on earth do they draw breath?
It is a brilliant biomechanical workaround. Since the shell is entirely rigid, ventilation of the lungs just cannot rely on the expansion of the chest wall. It's physically impossible.
So what do?
Instead, sea turtles rely on the massive muscles attached to their pelvic and shoulder girdles, as well as muscles anchoring to the plastron itself. They essentially use their limbs as biological bellows.
Biological bellows, that is so cool. When you watch footage of a female sea turtle hauling herself up a beach to nest, you often see this heavy, rhythmic, rocking motion with her front shoulders.
Yes, exactly.
And for a long time, I just assumed she was exhausted, fighting the crushing gravity that she usually doesn't feel in the buoyant ocean.
A lot of people think that.
But according to the Olive Ridley Project, that rocking isn't just a struggle to move forward, she is literally pumping her lungs.
She is. And the mechanics of that shoulder rock, as we call it, are phenomenal.
Walk us through it.
Okay, so because the lungs are attached to the dorsal interior of the carapace, so they're spread out along the top of the inside of the shell, the turtle has to change the internal pressure of its entire body cavity to pull air into them.
Since it can't expand its ribs.
Exactly. So by flexing the immense muscle masses around the shoulder and pelvic girdles, basically pulling them inward and then pushing them outward, they physically alter the volume of their internal cavity.
Oh, I see.
Pulling the shoulders outward drops the internal pressure, vacuuming air into the lungs, and then contracting them inward forces the air out.
So they're literally breathing with their armpits.
Basically, yes.
That is wild. The muscles that power their swimming strokes, the pectoral muscles, are pulling double duty as their respiratory system.
It's incredibly efficient.
But, breathing in the ocean isn't just about the mechanics of inflation. It's about surviving the dive itself. When they plunge hundreds of meters down, the water pressure is immense, and keeping that seawater out of the respiratory tract is quite literally a matter of life and death.
Oh, absolutely. And the head anatomy is specifically engineered for this watertight seal. Starting at the very front with the nostrils.
Right.
The NOAA manual details how the anterior tissue lining the nostrils is highly vascular and erectile. And in an adult sea turtle, this tissue isn't heavily innervated for smell like you might think. Its primary critical function is entirely structural.
Wait, okay. The idea of nasal erectile tissue immediately makes me think of human anatomy when we get a severe head cold.
Oh, that's a good comparison.
Like the blood vessels in our nasal turbinates swell up with blood, and it completely blocks our ability to breathe through our nose. It's an annoying immune response for us, but are you saying that for the sea turtle, that exact same physiological mechanism is a precision-engineered valve?
That is exactly what I'm saying. And the speed and reliability of that vascular response is incredible.
How fast are we talking?
Well, when the turtle is underwater, that tissue engorges with blood almost instantly, swelling outward to create a perfectly watertight, high-pressure seal against the ocean.
Wow. The Olive Ridley Project paints a really vivid picture of this in action. I want you to imagine a green turtle breaking the surface in the Maldives. The instant its snout clears the water, that erectile tissue rapidly deflates. The nostril just opens wide.
Right.
And then the turtle performs that massive shoulder pump we talked about, taking in a gasping, cavernous gulp of oxygen. And then, in just a fraction of a second before it plunges back beneath the waves, the blood rushes back into the tissue, swelling the nares tightly shut, locking the oxygen in and the crushing weight of the ocean out.
It is a perfectly choreographed sequence of pressure valves. And what's cool is this protection extends to their visual systems as well. The eyes are housed in these deep protective bony orbits in the skull. But, diving at speed means dealing with abrasive salt, suspended sand, microscopic debris.
It's not a pristine swimming pool.
Far from it. So they protect the globe of the eye with thick dorsal and ventral lids. But what's particularly interesting is that the ventral lid, so the bottom eyelid, is continuous with the conjunctiva.
That's the mucous membrane covering the front of the eye, right?
Correct.
That continuous membrane provides a really heavy layer of defense, but they also possess a nictitating membrane, right?
Yes, they do.
Which is so cool. It basically sits tucked away in the anterior and ventral corner of the eye, basically the lower front corner near the nose. And when they are swimming rapidly or moving through really abrasive environments, this membrane sweeps across the eye.
Like a windshield wiper.
Exactly. It acts like a built-in pair of clear swimming goggles, allowing them to maintain situational awareness while keeping the delicate cornea completely shielded.
So every sensory opening on the head is completely designed to slam shut or shield itself from the water.
Right, but this introduces the most fundamental paradox of marine life.
Which is?
Well, you can seal your nose and you can cover your eyes, but eventually you have to eat.
Yes.
And the moment a sea turtle opens its jaws to strike at a jellyfish or, rip a piece of seagrass, a massive volume of highly toxic salt water rushes right into its mouth.
And drinking that much seawater would cause massive osmotic shock.
Yeah.
The salt would just pull the water out of the turtle's own cells, leading to severe dehydration and kidney failure.
Exactly. Getting the food into the mouth is the easy part. The monumental engineering challenge is keeping the food moving down the digestive tract while simultaneously expelling the ocean water back out.
Okay, so how do they do it?
The entire architecture of their jaws and throat is designed around this single filtration problem. Let's look at the green turtle, Chelonia mitis. The NOAA manual breaks down the structure of its beak, which is called the rhamphotheca.
Rhamphotheca, got it. The outer layer of keratin is delicately built, especially when you compare it to, say, a loggerhead bone-crushing jaws. But the edges of the green turtle's beak are armed with heavily serrated spike-like cusps, supported by intervertical ridges along the bone.
So those cusps essentially act like a jagged pair of shears, perfect for gripping and slicing really slippery, fibrous seagrass.
Exactly.
But once that food, which is now accompanied by, a liter of seawater, moves past the beak, we enter what we described earlier as the biological sarlacc pit.
If we connect this to the bigger picture, the esophagus is really where the magic happens.
Yeah.
Because in most mammals, the esophagus is just a relatively smooth, muscular tube. It simply transports food to the stomach via peristalsis.
But not in the sea turtle.
No. In the sea turtle, the esophagus is an aggressive mechanical trap, The interior wall is completely carpeted in huge, conical, keratinized papillae.
And because they are made of keratin, which is the exact same protein as human fingernails or rhino horn, they are incredibly rigid and sharp.
Very sharp.
And crucially, every single one of these spikes points backward, down toward the stomach. Here's where it gets really interesting. The mechanics of how this works are awe-inspiring and honestly slightly horrifying.
It is a little gruesome.
When the green turtle swallows a jellyfish, the entire mass of food and seawater is forced down into this spiky cavern. And because the papillae point backward, the jellyfish slides over them pretty easily on the way down.
Right.
But then the turtle vigorously contracts the powerful muscles of its throat. It essentially squeezes its own esophagus like a tube of toothpaste from the bottom up.
And the sheer physics of that contraction forces the seawater back up and out of the mouth or out through the nares.
Right.
But the slippery prey cannot be pushed back out.
Because of the spikes.
Exactly. The backward-pointing papillae dig into the jellyfish or the seagrass, pinning it securely in place against the rush of the exiting water. It is a highly efficient, one-way biological filter that allows them to literally wring out their food before it ever hits the stomach.
It's incredible. And once the water is violently expelled like that, the papillae just abruptly end right where the esophagus joins the stomach, allowing the now compacted, relatively dry food to pass through smoothly into the acidic digestive chamber.
That's right.
But our sources highlight a really fascinating evolutionary divergence right at this specific junction.
Yeah, the regional variation is stark. It's really cool. If you perform a necropsy on an Atlantic green turtle, you'll observe that the spiked esophagus transitions smoothly and directly into the stomach lining.
There's no barrier or anything.
Right, there's no structural interruption at all. But if you look at a Pacific green turtle, you find a specialized, highly muscular crop situated right at the base of the esophagus, just before the stomach.
So it's an entirely distinct anatomical feature based purely on which ocean they swim in.
Yes.
Because a muscular crop-like in avian anatomy, for instance, is used for storing and mechanically grinding down tough fibrous food before the chemical digestion of the stomach takes over. So the presence of this crop in Pacific greens strongly suggests a deep evolutionary divergence driven by diet, right?
That's the leading theory, yeah. It implies that the Pacific populations are perhaps grazing on tougher, more robust species of macroalgae or seagrasses that actually require intense mechanical crushing, whereas the Atlantic populations might rely on softer forage.
Oh, fascinating.
So over millions of years, the Pacific turtles evolved an entirely new muscular chamber just to handle the roughage of their specific environment.
Wow. And the specialization doesn't even stop at the stomach. Even the roof of the mouth, before the food even hits the throat, is a completely complex sensory and respiratory junction.
Packed with structures.
Yeah, the palate contains internal, choani-internal nostrils that open directly above the glottis. And situated in the posterior lateral parts of the palate, so right near the hinge of the jaw joint, are the openings to the eustachian tubes, which lead directly into the middle ear cavity. Every single millimeter of space inside that armored skull is optimized to route air, filter water, or process food.
It has to be. And once that heavily siltered compacted meal makes it past the sarlacc pit and into the stomach, we enter the internal engine of the sea turtle.
Let's talk about the engine.
The digestive and organ layout beneath that rigid shell is an exercise in perfect spatial efficiency. When a biologist removes the peritoneum, which is the thin, tough membrane encasing the viscera, the organization is striking.
What does it look like?
Well, the heart sits centrally, just posterior to the trachea, and it's flanked by a massive, multi-lobed liver that takes up a huge percentage of the upper body cavity.
And the circulatory system weaving through this cavity is incredibly dense, right? Because it has to fuel the massive muscles required for swimming and breathing.
Yes, exactly.
The NOAA source maps out prominent vessels like the external jugular vein, which has to navigate through a really thick labyrinth of dorsal neck muscles, specifically the biventer cervical and transverse cervical muscles.
Right.
And these are the heavy cables of muscle that allow the turtle to explosively retract its head or strike at prey.
And routing that blood flow under a rigid, immovable shell requires precise anatomical pathways.
Because they can't just expand or shift around easily.
Exactly. The manual highlights the epigastric vein, which is a major vessel that travels dorsally to supply the extensors and flexors of the hind legs. It runs perfectly tucked, just medial to the marginal scutes of the carapace.
So it's routed almost like electrical wiring along the baseboards of a house.
That's a perfect analogy.
Keeping the delicate vessels protected by the thickest parts of the armor, while still supplying the powerhouse muscles of the pelvis.
And those deep ventral hip muscles, the adductor femoris and the pubic femoralis internus, are the true engines of their agility.
Because they don't have spinal flexibility.
Exactly.
Yeah.
In an animal with no spine to bend, these muscles act as massive antagonists, pulling against the unyielding anchor of the plastron to generate the torque they need to pedal their hind flippers and actually steer.
But supplying all this muscle with blood creates metabolic waste, which brings us to perhaps the most critical organ system for a marine reptile.
The kidneys.
Yes!
Because even with the spiked throat filtering out the bulk of the seawater, the turtle still ingests a significant salt load through its food and just the residual water it swallows. The osmotic pressure on their internal cells is constant.
So to survive, their kidneys basically have to function as a high-capacity desalination and waste management plant.
Absolutely. The NOAA manual describes these kidneys as paired, lobular, elliptical red structures. But what's really crucial here is their physical location.
Where are they?
They are located retroperitoneally.
Okay, meaning what?
Meaning they aren't floating freely in the abdominal cavity with the stomach and the liver. They are tucked firmly behind the peritoneum, squished and adhered right up against the underside of the carapace.
Ah, okay. So that retroperitoneal placement is really a brilliant spatial adaptation. By pinning the kidneys to the ceiling of the shell, outside the main peritoneal sac, the turtle frees up massive amounts of real estate in its main body cavity.
Real estate that is absolutely necessary.
Right, for the expansion of their enormous digestive tracts, which have to ferment huge volumes of seagrass, and for the massive expansion of their reproductive organs during breeding season.
Precisely. And the manual also specifically classifies sea turtle kidneys as metanephric.
Okay, let's unpack this word, metanephric. Treat it like an architectural blueprint for us.
Okay, so we're talking about their specific embryonic origin here. They arise from the posterior nephric ridge during development. But functionally, what this means is their internal plumbing is highly advanced compared to simpler vertebrates.
Advanced how?
The kidney tubules are drained by dedicated, robust ureters. These are thick tubes designed to rapidly transport the concentrated salty waste away from the organ.
Right, because they have to handle the extreme osmotic pressure from living in salt water.
Yes. And all of that internal plumbing, the ureters managing the salt waste, the immense digestive tract processing the food, and the gonadal ducts managing reproduction, all of it must eventually exit the armored shell.
And they do this through a single master valve, the cloaca.
The cloaca is the ultimate common chamber.
It's multitasking at its finest.
It really is. The rectum from the digestive system, the ureters from the kidneys, the gonadal ducts, and the urinary bladder all empty into this single vent.
And the bladder itself in a sea turtle is fascinating because it doesn't just hold waste, does it?
No, it functions as a critical reservoir for storing water and managing internal fluid balance. It acts as a buffer against that salty environment.
The efficiency of having a single anatomical exit point in a heavily armored animal just cannot be overstated. And it is through this same chamber that the immense biological cost of reproduction is paid.
And that cost is steep.
Very steep. The Necropsy manual gives us a stark, visceral understanding of how heavily a sea turtle invests in creating the next generation.
Dr. Werg's manual details the visual transformation of the female reproductive system, and the contrast is just striking.
Walk us through it. What does a biologist see?
Well, the gonads are attached to the peritoneal wall, anatomically ventral to those retroperitoneal kidneys we just talked about. If a biologist examines an immature, juvenile female turtle, the ovaries are visually unremarkable.
Like, they don't look like much.
Right. They just look like a simple, flat, pink ribbon of tissue with a slightly granular texture. And the oviduct, which will eventually carry the eggs, is just a narrow, flat, translucent tube.
So it's basically a dormant system taking up minimal space and energy.
Exactly. But when that female reaches sexual maturity and prepares for a nesting season, the transformation is explosive.
Explosive is the word.
Those flat pink ribbons develop massive, round, yellow follicles. And depending on the stage of development, these follicles range from 2 millimeters to 2 centimeters in diameter.
Which is huge. The manual describes them looking like vibrant, heavy, pearl-like strings of yellow spheres embedded throughout the entire organ. Just picture that transformation from a tiny pink ribbon to these massive yellow spheres.
The sheer metabolic energy required to generate hundreds of these yolk-filled follicles is staggering.
And the oviduct has to change too, right?
Yes, it undergoes an equally dramatic structural change. To accommodate the passage of these massive follicles and to lay down the albumin and the leathery eggshell before they reach the cloaca, that tiny flat tube hypertrophies.
It grows.
Massively. It widens and develops heavy folds, eventually looking exactly like a thick biological accordion.
A biological accordion. The spatial mechanics inside the shell become incredibly tight during this period, I imagine.
Oh, extremely tight.
You've got the lungs, the massive liver, the fermenting stomach, and now these accordion-like oviducts swollen with hundreds of eggs, all confined within an unyielding bone box. It really is a biological pressure cooker.
It is. And, the precision of these anatomical details, knowing the exact texture of an immature ovary versus the precise retroperitoneal placement of a mid-and-effort kidney, it's astounding.
It is.
But this raises an important question. How do we actually acquire this knowledge?
Right, because we aren't just observing these animals in pristine, perfectly controlled laboratories.
Not at all. They spend the vast majority of their lives vanishing into the pelagic depths of the open ocean.
Which brings us to the critical and often incredibly unglamorous field of marine pathology. Because to truly understand how these magnificent animals live, and to understand the threats pushing them toward extinction, we have to meticulously study how they die.
Yes. Which brings us back to Dr. Thierry M. Work's Sea Turtle Necropsy Manual for Biologists in Remote Refuges.
Okay, let's unpack this manual, because reading this completely shatters the romanticized illusion of marine biology.
Oh, it really does.
We aren't talking about swimming alongside sea turtles over beautiful coral reefs in a sleek wetsuit. We are talking about a biologist standing on a sweltering, humid beach, hundreds of miles from the nearest hospital, armed with a bone saw, a scalpel, and industrial chemicals, just trying to crack open a rigid shell to find out what killed the animal.
It is brutal work. Dr. Work designed this manual specifically for field biologists who may actually have very little formal training in veterinary pathology.
Really?
Yeah, these are often conservationists who just find themselves as the absolute only line of defense in incredibly isolated locations. So he emphasizes safety above all else because the stakes are incredibly high.
And the warnings in the manual are so stark, but obviously necessary. He literally writes, "Doing a necropsy on a turtle is hard work. Be careful with knives and sharp bones and follow proper hygiene. Do not eat or drink while dissecting a carcass."
He seems absurd, right?
It does. It seems absurd that you would have to explicitly tell a scientist not to eat a sandwich while sawing through a decaying sea turtle. But when you are exhausted, severely dehydrated, and working a 10-hour day on a tropical atoll,
Cognitive fatigue sets in.
exactly, people make mistakes.
And the risk isn't just a simple slip of the knife. He repeatedly warns about zoonotic diseases.
Diseases that can jump to humans.
Exactly. Pathogens that can jump from the decaying marine reptile directly into the human circulatory system through just a tiny cut on the hand. It is hazardous, high-stakes detective work.
So the core methodology of this detective work relies on establishing a really rigid baseline of what is normal so that the biologist can actually spot the abnormal.
Right, because if you don't know what a healthy internal engine looks like, you cannot diagnose the failure.
The manual uses the liver as a prime example of this. The liver of a healthy green turtle is massive and visually very distinct. Dr. Work states it should be firm to the touch, with smoothly rounded borders, and display a homogeneous, rich, dark purple-brown color.
That baseline is essentially the biologist's compass.
Right.
When they open the peritoneal cavity, they immediately assess the liver against that standard, and the deviations become the clues.
Like what kind of deviations?
Well, the liver shows spots or blotches of pale yellow or green. That indicates a systemic failure.
Okay.
And the physical consistency is equally important. If the liver tissue feels mushy and overly soft, or conversely, if it is rigid and hard like a piece of wood, it indicates massive cellular damage, fibrosis or necrosis.
Wow.
The shape might even be warped by lumps, nodules, or deep fibrous scars from past parasitic infections.
But identifying the abnormality is only step one. The crucial task is capturing that evidence and preserving it so it can survive a journey of thousands of miles back to a laboratory for microscopic analysis.
And the rules for chemical preservation, even when you're standing on a sandy beach, are utterly unforgiving.
To perform formalin fixation, which is the process where the chemical formaldehyde cross-links the proteins in the tissue, stopping decay and preserving the cellular architecture for a pathologist to examine under a microscope, the biologist must adhere to strict physics.
You cannot simply hack off a giant chunk of liver and throw it in a bucket of formalin.
No, the manual dictates a very strict volume ratio. There must be an absolute minimum of 2 parts formalin to 1 part tissue.
But the geometry of the tissue sample is actually even more critical than the volume.
Yeah, how thin does it have to be?
The slice of organ can be no thicker than 0.5 centimeters.
Half a centimeter. That's incredibly thin.
It has to be. The biochemistry of fixation dictates this rule. Formalin penetrates tissue at a very specific, slow rate.
Okay.
If the biologist cuts a piece of liver that is, say, an inch thick, the formalin will fix the outside layers, creating this hardened chemical shell around the tissue.
But it won't reach the center.
Exactly. The chemical will not reach the center of the tissue fast enough. So the inside of the sample will literally rot and liquefy from the inside out, completely destroying the cellular evidence before it ever reaches the lab.
That's tragic after all that work.
It is. Furthermore, the manual explicitly forbids freezing any tissue destined for formalin.
Why is that?
Because the ice crystals will physically puncture and shred the delicate cell walls, rendering microscopic analysis completely impossible.
But I think the most brilliant, real CSI-level instruction in the sampling protocol is where the biologist is instructed to make their surgical cut.
The margin.
Right. When they locate a diseased lesion on an organ, they don't just cut out the center of the rot, they are instructed to capture the margin. They must carefully slice out a piece of tissue that contains half of the abnormal diseased lesion and half of the visually healthy tissue immediately adjacent to it.
Because capturing that margin is exactly like taking an aerial photograph of a battlefield.
I love that analogy.
The margin is the active front line where the turtle's immune system is violently clashing with the invading pathogen. When the pathologist stains that slide back at the lab and looks at it under a microscope, the margin reviews the entire story.
It shows the battle in progress.
Yeah, they can see the specific types of white blood cells rushing to the defense. They can see how the surrounding healthy cells are reacting or dying. And they can often identify the specific bacteria, fungus, or parasite leading the assault.
And alongside the formalin samples, the field biologist must also collect frozen samples, about 20 to 30 grams of tissue, wrapped in foil or placed in Whirlpack bags. But the manual stresses that these must be collected as early as possible in the necropsy process, ideally the moment the shell is opened.
Yes, and this speed is required to prevent cross-contamination.
From the gut.
Exactly.
Right.
Once you open that massive fermenting digestive tract, the bacteria from the gut will rapidly spread across the entire body cavity. If you want to isolate a specific microorganism that caused an infection, or detect trace amounts of heavy metals or chemical toxins in the liver, you need pristine, uncontaminated tissue frozen immediately to halt any bacterial growth.
But the reality of remote fieldwork is that resources run out.
They always do.
What happens when a biologist is stationed on a remote Pacific island? The supply plane is delayed by 2 weeks, a massive green turtle washes up dead, and they are completely out of properly buffered formalin. Because the chemical buffer is required to keep the formalin at a neutral pH so it doesn't degrade the tissue?
This is where Dr. Work's manual shines as a testament to pure scientific ingenuity. He provides a brilliant, albeit desperate, field hack.
Let's hear it.
If you lack commercial buffered formalin, you can synthesize an adequate substitute by mixing 15 parts of raw, 37% formaldehyde, with 85 parts of local seawater.
They use the ocean itself as the chemical buffer.
They do. The natural alkalinity and mineral content of the seawater stabilizes the harsh formaldehyde just enough to preserve the cellular structure.
It's brilliant, but it sounds deeply hazardous.
Well, it is. Dealing with raw, unbuffered formaldehyde on a hot, windy beach while wielding a bone saw is terrifying work. The manual explicitly mandates heavy gloves, working upwind in well-ventilated areas, and aggressive hand washing. The fumes alone can be toxic.
The logistical discipline required in these environments is just phenomenal. The manual even dictates the specific type of writing instrument used to label the jars.
It's that specific.
It is. All labels must be written in pencil or indelible ink, like a Sharpie. Ballpoint pens are strictly forbidden.
Because of the environment.
Right, the humidity, the seawater, or even formalin splashes will cause ballpoint ink to just run, completely erasing the data. And they are even instructed to abbreviate the month with letters like MAR for March instead of just the number 3 because international labs use different date formats.
Oh, that makes sense. Month, day, year versus day, month, year.
Exactly, and a simple misinterpretation of a date can ruin the entire epidemiological timeline of a disease outbreak. Every single action, every half-centimeter cut, every pencil stroke on a label is an act of sheer dedication. The biologist is fighting heat, exhaustion, and decay to preserve a microscopic clue that might travel across an ocean to a lab, all in the hope of understanding what is killing these animals and how we might stop it.
And the depth of our knowledge about the sea turtle's internal engine, the metanephric kidneys, the massive shoulder muscles pumping the lungs, the sarlacc pit in the throat, it's built almost entirely on the backs of these field scientists executing these grueling necropsies in the most unforgiving environments on the planet.
Which brings us to the profound reality of what we've unpacked today. We have deconstructed an animal that has pushed the vertebrate body plan to its absolute breaking point just to survive in the ocean. We started with the foundation, seeing how they traded the flexibility of a spine for an impregnable fused dome of dermal bone.
Right.
We explored the brilliant biomechanical workaround of using their immense pectoral swimming muscles to pump their lungs against a rigid shell.
And we examined the high-pressure valves of their head anatomy, the erectile tissue inflating in the nostrils to seal out the crushing depths, and the nictitating membrane sweeping across their eyes.
And we mapped the highly efficient internal plumbing from the retroperitoneal kidneys fighting a constant war against osmotic pressure to the ultimate multi-purpose common chamber of the cloaca.
But we also marveled at the terrifying efficiency of their esophagus.
The sarlacc pit.
Those massive backward-pointing keratinized spikes lining the throat. It is an adaptation perfectly honed over millions of years of evolution. It allows the sea turtle to violently contract its throat, expelling toxic seawater back into the ocean, while the daggers trap the slippery, gelatinous body of a jellyfish, holding it firmly in place before it is swallowed into the stomach.
It is, without question, a biological masterpiece.
It is a flawless mechanical trap, but it is fundamentally a one-way street.
Yeah.
Once an object moves past those backward-pointing keratin spikes, the very architecture of the throat dictates that it cannot be regurgitated, it cannot come back up.
Which leaves you, the listener, with a chilling mechanical reality to consider. What happens when an animal equipped with an inescapable one-way trap in its throat encounters a clear plastic grocery bag drifting in the water column? A synthetic object that looks exactly and moves exactly like the gelatinous bell of a jellyfish. Evolution spent millions of years building the perfect anatomical filter to exploit a specific food source, but in doing so, it left the sea turtle tragically vulnerable. Humans may have inadvertently created the perfect lethal hazard to exploit a biological masterpiece. It's definitely something to consider the next time you see a piece of plastic blowing across the sand toward the water.